So hello, everybody. Thank you very much for joining us again. So welcome to our Center for One Health Annual Conference and to our session on emerging pollutants. So briefly, as you may know by now, the format of the session will be as follows. So we're going to start with a 30-minute talk by our plenary speaker. And then her talk will be directly followed by 3 10-minute talks from researchers from our Center for One Health here at NUI Galway, and they are all working on the theme of emerging pollutants. So at the end of the talks, we will have a 30-minute Q&A session where you will be able to ask all of your questions to all the speakers. So we ask all attendees to put your questions in the Q&A box and not in the chat, please. So I am absolutely delighted to welcome our keynote speaker, Professor Cynthia Duvet, who is a professor in environmental science at Stockholm University. And Cynthia first started working on organic environmental contaminant while she was conducting a national survey of polyfluorinated dioxins and related chemicals, and she did that back in 1988. And at the time, Cynthia was working with the Swedish Environmental Protection Agency. Then Cynthia moved to Stockholm University in 1992, and her recent research has focused on legacy and emerging flame retardants. So that includes brominated, chlorinated, and organophosphate-based contaminants. So Cynthia is interested in their fate in indoor and outdoor environments, as well as human exposure, and she is interested in both adults and also children, and the effect, of course, that those compounds have on exposure. So in addition to working on terrestrial ecosystems, Cynthia also investigated the fate of contaminant in seafood webs. So Cynthia is also a co-lead of the Persistent Organic Pollutant Expert Group of the Arctic Monitoring and Assessment Program since 1994. And in that capacity, she has led 5 international assessment reports on persistent organic pollutants, including contaminants of emerging concern and in the Circumpolar Arctic. So today, I'm absolutely delighted that Cynthia is with us, and she's going to tell us about exposure and effect of legacy and emerging persistent organic pollutants. So over to you, Cynthia, whenever you're ready.
Okay. So I hope you can see my screen now. Yes, thank you for inviting me to this interesting session. I was in Galway a couple of years ago before the pandemic broke out, and held something, a similar type of presentation there. I'm going to talk a little bit about legacy and emerging persistent organic pollutants and exposure and effects. And I'll give a little bit of background, a bit of history of these chemicals, what they are, something about exposure and how this occurs and effects that we know from some of these chemicals, and then cover some of the new and emerging POPs. And the history goes back to World War I. This is the development of this modern synthetic chemistry industry was during this period. This is when countries put together chemical industries to create chemical weapons like mustard gas and phosgene. And after the war, after having laid this groundwork for synthetic chemicals industry, they started to use this capacity to create new chemicals that could be used for the betterment of human society. So they created new chemicals like polyfluorinated biphenyls, which were first manufactured in 1929. Nylon, the very first synthetic textile, was produced in 1938. DDT, a revolutionary pesticide used for agriculture, was developed in 1942. So these chemicals were seen as a really positive side of the chemical industry and the synthetic chemicals industry. But in the 60s, 50s and 60s, the backside of some of these chemicals became apparent. In particular, some of these pesticides that were being used, they were being spread widely in the environment, and suddenly they were not only killing the insects that they were supposed to be killing, they were also killing other animals like birds and birds of prey. And DDT was one of the first of these synthetic organic toxins that was found in the environment. And Rachel Carson wrote her book, Silent Spring in 1962, which was actually a science, in a sense, a popular science expose of all these pesticides and what they were, the damage they were doing in the environment. So this was the backside of these chemicals. And so at some point they started to be called persistent organic pollutants. And this first wave, all were organic compounds containing chlorine. And so they're often called organochlorines. Classics, of course, were DDT and the polychlorinated biphenyls, but there were also other various pesticides like hexachlorobenzene, the DRINs, dieldrin, aldrin, endrin, chloridane. And then another group popped up later called the polyfluorinated dioxins and furans. We'll look a little more closely at them because they weren't produced intentionally. So if we look at some of these chemicals, just to see what their properties are and why they were so, why they have become such a problem. This is DDT, and what is often a common denominator with these chemicals is that they have a ring form on them, one or two of these kinds of benzene rings, and then they have chlorine off of them. So here we have two benzenes and the chlorines. So these are organic compounds with chlorine. PCB or DDT is very persistent. It lasts a long time, which was really good in agriculture because that meant, of course, you didn't have to spray as often to kill pests. It's not acutely toxic to humans. And as an insecticide, it was widely used in agriculture. It was used in pest prevention to get rid of flies. And nowadays, it's only used for malaria prevention to kill the malaria mosquitoes. For PCBs, the polychlorinated biphenyls, again, we have a benzene, two benzene rings connected to each other, and this can contain anywhere from 1 to 10 chlorine molecules off the rings here. These are very stable compounds. They're not acutely toxic to humans. They're produced in mixtures. They tolerate high temperatures without burning, and they lead heat and electricity well. And this made them perfect for use in electrical transformers. So PCBs were critical to actually electrifying society because you would have these transformers filled with this oil, and they would then lead the electricity so that it jumped from transformer to transformer through the electric wires and send electricity into our homes. But PCBs were also kind of miracle chemicals. They could be used for a lot of other applications. They were used in condensers. They could make plastic soft. So they were used as plasticizers in plastics and paints and in caulking. They were used in self-copying paper, and they were also even used as flame retardants. The polychlorinated dibenzodioxins and furans, again, we have these benzene rings, but now they are connected, in this case, with two ether bridges, an oxygen here, or one ether bridge here for the furans. And then you have the chlorines. And this is the most toxic, 2,3,7,8 tetrachlorodibenzodioxin. And these are very toxic chemicals, and they are byproducts. They are not actually intentionally made by us synthetically. They are byproducts that accidentally get produced when you make PCBs. They are produced when chlorine is used to bleach paper and pulp. So they were released around paper factories. And the chlorine production also around the chloralkali plants that created the chlorine for the bleaching. They're also created when you incinerate waste. So waste incinerators in the 80s and 90s were major sources of dioxins to the atmosphere. And they're also accidentally produced when you make chlorinated phenols or chlorophenol production. And these could be things like Agent Orange, which was used as an herbicide. So dioxins were mixed, in a sense, in these chlorophenol mixtures that were used widely, for instance, in the Vietnam War. For The dioxins and furans, one of the biggest accidents that occurred that included humans was in Seveso, Italy in 1976. And there was a trichlorophenol factory that had a runaway reaction, and it exploded. And it released a cloud of pure tetrachlorodibenzodioxin into the town. And shortly thereafter, within days, animals started to die, cats, birds, dogs, horses. And when the government realized what had been released there, they had to evacuate this town. And a long-term health study of the occupants started. And here you can see what's called chloracne, a child who was exposed, highly exposed to this TCDD. This is one of the major symptoms that you see in humans that have been exposed to this. It's a kind of an acne or outbreak on the skin. And here you just see where they have access, they're putting up the signs telling people not to come into the city. So for POPs, these persistent organic pollutants, the major sources were industries. They were point sources. Industries releasing these chemicals directly to air, water, and sediments in the nearby environment. They were used in agriculture, so the widespread spraying of agricultural crops. They were coming from waste incinerators and being released into the air. And they were being used in these transformers. So they were outdoors in transformers that possibly leak these chemicals into the environment. So most of the release was predominantly to the outdoor environment. So what happens to these chemicals once they're released into the outdoor environment? Well, there are a couple of properties that these chemicals have that tell you where they're going to go. They hate water, they're hydrophobic. And that means that if they are released into water, they're going to look for an organic phase. And in the environment, this is either organic carbon or it's fat or lipids. These act as solvents. They're also semi-volatile. That means that they don't readily evaporate. So if they're released into the air, they don't like being in air. They want to be in organic carbon or fats. So they will move from air into an organic phase. But a little bit will still be in water and a little bit will still be in the air. So they actually distribute between these two phases. So in the environment, where they will go is into the organic carbon that you find in soil, in suspended sediments and bottom sediments, and in aerosols, so on particulate matter that's in the air. And when it comes to, for instance, in water, they will be also looking for fat sources in organisms. So for instance, in fish, they will be entering the fish and essentially accumulating in their fats. So in the 70s and the 80s, because these chemicals were being found after Rachel Carson's book came out and people started looking for them everywhere, there was a lot of research done in the Great Lakes and in the Baltic Sea, which were highly industrialized water bodies. And high concentrations were being found of these POPs in fish and birds. And these were coming from direct sources because you have a lot of chemical industries, paper and pulp mills and the like along these. But in the late 80s, in Canada, scientists started to also find PCB in the high Arctic, far away from these kinds of direct sources. And the question, of course, was where was this coming from? How could these chemicals actually be reaching the Arctic? If there were no direct sources, it must mean that they're somehow transported up there. But what would explain that? So here is a circumpolar perspective on historical use of PCBs. And what you can see is that most PCB usage was in the eastern part of North America, in Western Europe, and in Japan. There was virtually no PCB usage up in the high Arctic, but it was being found in seals and polar bears and in humans as well. And scientists studying the atmosphere were understanding now that air pathways actually passed right over these source regions and delivered these chemicals up into the atmosphere, up into the Arctic. So they're coming in air over these source regions and then carrying small molecules or small amounts of these chemicals up into the Arctic. So as I told you, they're semi-volatile, which means some of them will still, some molecules will still be in the air. So this could explain how they were being delivered to the Arctic. But then how are they getting into the ecosystem? And here it was some modelers in Canada who posited that you might be seeing something called global condensation. And this is based on temperature and volatility. You think of any kind of chemical or even water, if you raise the temperature, you increase evaporation. And high temperature then is something that increases the volatility of a chemical. If you lower the temperature, you decrease its volatility and it will condense. And this is what happens, for instance, when you're on a hot summer's day and you take out a cold can of, for instance, soda pop, and the can gets little droplets of water. And it's because the water that's in the air is evaporated, but when it hits the cold surface, it condenses out. And pops behave this way in the environment. And what they found was that if you use these chemicals in the southerly latitudes, for instance, in the tropics or in our parts of the world, it's warmer and they will evaporate and they will travel in the air. And then when they hit the cold air of the Arctic, they will condense out. And what they essentially hypothesized is that even these chemicals that have relatively low volatility will, to some extent, be evaporated. They will move north. They will hit cold air, for instance, in the winter. They will condense out. Then the summer will come. It will get warm and they will re-evaporate or re-volatilize. And then they will continue their journey northwards until they hit cold air again and condense out again. So that some chemicals like DDT and Pcb can hop, they call them multi-hoppers, slowly but surely up into the Arctic. And chemicals that are have higher volatility will be able to actually jump to the Arctic maybe in one go. So this would explain how these chemicals were actually getting into the ecosystem. So summary for the organochlorine pops is that they were in widespread use. They were released to outdoor environments by point sources. High levels of pollution were found near sources, for example, in the Baltic Sea and the Great Lakes. They're stable, they're persistent, and that means that they can travel long distances in the atmosphere. They don't break down very quickly. And we find these chemicals now all over the globe. We find them in the Arctic. We also find them in Antarctica. So then the next step is, how are they getting into organisms? Why are we finding these chemicals in polar bears? And to understand this, you need to understand how chemicals move up food webs and the difference between short and long food webs. And a terrestrial food web is very short, often a very short food web. You have grass that's eaten by a cow and we eat the cow. So that's 2 steps, 1, 2. In aquatic food webs, you can have somewhat longer food webs. You can have plankton that are exchanging with the water. Then you have fish that eat the plankton. A bigger fish eats the smaller fish, and a fish-eating bird eats the big fish. So you have 1, 2, 3 steps. In the Arctic, you have 2 things. You have long food webs, but you also have a very fat-rich diet, in a sense, because most of these animals have to live off of fat during the winter, or they have to have thick fat reserves to protect them from the cold, like blubber. So even plankton in the Arctic have more fat in them than plankton further south. So they can sequester or suck up like a sponge more of the pops that are in the environment. And then there are many steps, and you can get, you can get up to 4 to 5 steps. And polar bears and humans are actually at the top of this food web, because there are indigenous populations that eat marine mammals and actually eat polar bears as well in the Inuit communities, for instance, in Greenland. So you can end up with up to 4 to 5 steps, which gives these chemicals a much longer pathway up into the top of the food web. So what happens? Well, these chemicals are fat soluble, and it leads to enrichment up a food web. So for each food web step, the concentration will increase approximately 3 to 10 times. It will magnify. And that means that when you have areas with high concentrations from point sources, but a short food web, you can get very high levels, for example, in the Baltic Sea or the Great Lakes. Or if you have a very long fat-rich food web, you can start with lower concentrations, but still end up with very high concentrations at the top, for example, in the Arctic. So just to illustrate this, this is a food web in the Baltic Sea from zooplankton to herring to gray seal. And you can see that the chemicals increase 5 times up to the herring and then even more going from herring to the gray seal. But you end up with about 20,000 nanogram per gram lipid in the gray seal. In the Arctic, in this case, it's a 4-step chain from zooplankton to polar cod to ring seal to polar bear. You start out with a lower concentration in the zooplankton, but you still end up with about the same concentration in the top predator because it's a longer food chain. Human and wildlife exposure then to these legacy pops has mostly been via dietary intake. And for humans and also for many of the marine mammals and fish-eating birds, this has been from fatty fish, in particular like trout and herring and salmon, the salmonids. They have higher fat contents in their tissues, so they then can essentially contain more of these contaminants in their muscle tissues. And there were high exposures along the Great Lakes and in the Baltic Sea populations, both for wildlife and humans. And this has actually led to fish consumption guidelines in many countries and guidelines for protecting fish-eating organisms. And for the WHO, the World Health Organization, they created tolerable daily intakes for some of these chemicals, such as for dioxins, and in some cases have actually, for instance, in the EU, forbidden the sale of some of these fish species if they come from the Baltic Sea, for instance, because of the high dioxin levels. If fish has not been an important food item in some populations, then meat and dairy products usually are the major source for a lot of these chemicals. But in the Arctic, you have certain populations that are mostly impacted by marine mammal consumption. And this has led to the highest PCB levels in the world in Arctic indigenous populations. The Inuit, for instance, in Greenland have higher levels, and even in Canada here, that we have these very high levels in humans. And this is still the case. PCBs are still higher, essentially, in many of these Arctic communities. If we look at DDT, on the other hand, it's a little bit different story. This is a time trend going back to the 60s of DDT concentrations. This is from the World Health Organization and UNEP combined. They give out these, they do these studies following temporal trends after the bans of many of these chemicals. And you can see that for the northern general population, which is North America and Western Europe, the concentrations have gone down from a very high level. And these populations have mostly been exposed via long-range transport and diffuse emissions, and getting into the food web and ending up in the food chain. For the Inuit, the indigenous populations in northern highly exposed populations here, you can see that they started out quite high because of their marine mammal consumption, and it's still quite high because of their marine mammal consumption. In some communities, it has gone down where they have stopped eating as much of the marine mammals, gone on to other foods. But you see that the very highest levels now are in the tropics. And this is because these are areas where DDT is being sprayed locally to fight malaria. And so when they come and spray around the doors and windows of your home, of course, your food, anything in your house will be contaminated. And that means you will be much more highly exposed. Effects. Well, the first effects of POPs were seen in wildlife, particularly in the Great Lakes and the Baltic Sea. There were basically global population declines, especially in birds of prey and in colonial water birds. And this was mostly due to eggshell thinning from DDE or DDT exposure. DDE is the metabolite that it turns into. A lot of bird species were also found to have reproductive impairment with high embryonic and chick mortality. You could see these deformities with crossed bills, and they had club feet, edema and decreased body weight. And this was linked to dioxin like Pcbs and dioxin exposure. In mammals, the studies were mostly in mustelids like mink and otter, and in seals, and they saw reproductive effects. Ranched mink that were being fed Great Lakes fish, for instance, in the 70s had total reproductive failure. There were population declines of wild mink and otter in the Great Lakes and in Europe, and they're only, especially the otter, are only now coming back. Effects in wildlife. These wildlife were linked to the Pcbs and the dioxins. And for the Baltic Sea seal population, there were huge declines in the 60s. They were on the way to being extinct. There were high incidences of aborted seal pups. When they opened these seals up, sometimes they could find occlusions in their uterus so that they couldn't actually get pregnant. There were various other deformities, and these were all linked to very high PCB concentrations. And for people, many of these chemicals were found later to be carcinogens. They lead to reduced immune systems. So Inuit populations, for instance, children have higher infection rates where they have the highest PCB levels. Neurobehavioral effects have been seen in highly exposed children. These have both been shown in the Great Lakes and in some Arctic studies. Lower birth weights, shorter pregnancy, effects on men's reproduction. And in Seveso, they saw skewed sex ratios, that fewer boys were born in the generation after the Seveso accident, in the parents that were exposed as young people in that accident. So we call these the legacy POPs. Their effects were seen mainly in highly exposed populations, wildlife and humans. The early life stages are the most sensitive to exposure, for instance, either in the embryo stage or in the uterus. These are called persistent bioaccumulative and toxic chemicals, or PBT chemicals. They undergo long-range atmospheric transport and can transport all around the world. And in 2004, the UN Stockholm Convention on POPs came into effect and banned or restricted 12 of these, including DDT, PCBs, and dioxins. So after these chemicals came a second generation of POPs. And many of these contain not just chlorine, they may contain bromine or fluorine. These are, for instance, brominated flame retardants and per and polyfluorinated substances. They have similar characteristics to these legacy POPs. Many of them are hydrophobic and semi-volatile, and they have a similar environmental behavior to legacy POPs. So just as examples, here are the polybrominated diphenyl ethers. These have been used as flame retardants. Again, we have our rings, we have an ether bridge, but now we have bromine instead of chlorine. And there were 3 technical products. Penta BDE was used in soft foam in furniture and in textiles, and Oct and Deca have been used in hard plastics, for instance, in computers and televisions, and in the back coating of textiles in cars, planes, and trains. The per and polyfluorinated alkyl substances, the most famous is Pfas, perfluorooctane sulfonate here, are, instead of rings, they are a chain, an alkane. So it's a carbon chain of, in this case, 8 carbons that is fluorinated. And this is an extremely strong bond, and this makes this extremely protected. So these chemicals are sometimes called the forever chemicals. They do not break down in the environment. But what's unusual about these is they have this little head here, which actually is ionizable. So this part is hydrophobic, but this part is actually possible to dissolve in water. So these chemicals actually travel better in water than other POPs. So this has caused other problems, which we'll get into. But these have been used as surface treatment for clothing, textiles, paper, because they repel, this hydrophobic part repels water, oil, and dirt. It's also been used as a fire extinguishing foam, for instance, in aviation and in airports. And this has caused problems recently because they have now found it in groundwater because it's been leaking from these airports. Another group is the Perfluorocarboxylic Acids, and PFOA is the most famous of these. It was used in the production of other PFAS and polymers such as Teflon in Teflon pans and Gore-Tex, which is used in clothing and in shoes. So the sources for these new POPs are still direct sources. We still have production facilities to outdoors, and in many cases, waste facilities where they dumped wastes have turned out to be pollution sources. But indirect sources now are also important because these chemicals are in consumer products we use all the time, and we use them indoors. So that means we can be exposed to them from the products indoors, building materials that are used indoors, and then again from this groundwater contamination near airports, which is causing a problem for drinking water supplies. New exposure routes, well, diet is still important, and especially for wildlife, but drinking water now is an issue for the PFAS. And indoor exposure for humans is coming from, for instance, dust ingestion, air inhalation, hand-to-mouth activity, and dermal uptake even for the flame retardants as well. So the second generation of POPs have been found to be endocrine disruptors in many cases. Some of them have PBT characteristics and have been banned or restricted. And these include all three of the technical products of the polybrominated diphenyl ethers, hexabromocyclododecane, and PFOS and PFOA. Effects that have been seen are similar as those seen for the organochlorine POPs. There are immune effects, reproductive effects, they're carcinogens. For many of the BFRs, they affect the thyroid in particular, so thyroid hormones. They cause reproductive effects and behavioral effects. And in children, there have been several epidemiological studies now showing neurobehavioral effects and measurable decreases in IQ in highly exposed children. For PFAS, there's a lower antibody response in children after vaccination that is directly related to PFOS concentrations, which has led the European Food and Safety Authority to recommend reduction of the tolerable daily intake of PFOS by 6 recently. And these PFAS are also carcinogenic. And now we have, in a sense, this new generation of emerging POPs being replaced by even more similar chemicals, a third generation of chemicals or emerging POPs. And these again have either bromine or fluorine, and in some cases are now chlorinated again. We have chlorinated paraffins as a big new group of chemicals in commerce. And we also have things like organophosphorus, flame retardants that are being used. And we call this regrettable substitution, that we ban a chemical, we replace it, that chemical gets banned, and then we replace it again. And the replacements always seem to have similar enough chemical properties that they behave like POPs. So here are just a few of these new brominated flame retardants. There are something like 25 to 30 of these that are replacing the PBDEs, the 3 technical products. And organophosphate esters are also being used to replace a lot of the old brominated flame retardants. And the PFAS, we had PFOS and PFOA. Now there are as many as 3,000 PFAS on the global market that are replacing those substances because of their desirable properties. And if we look at global chemical production, essentially it has exploded, going from 7 to 400 to predicted 1,200 million tons in 2050. And here you can just see this is the synthetic chemical change as trends in global trade value as a proxy for production. You can see how global chemicals, pesticides, and global chemical industry output is essentially increasing exponentially at the moment. So in conclusion, the legacy POPs, there were not that many of them, but the exposures were quite high. But the new and emerging POPs, there are more of these chemicals, but often the exposures at the moment are lower than they were to the old POPs. But this raises some questions that we have mixture effects. We don't know what all of these chemicals together have in the way of an effect. And that we are now also being exposed from indoor media and products, not just diet. And that becomes a new exposure source that we don't really understand very well. And then we have a new and growing number of chemicals in consumer products and a lack of data on effects in humans and wildlife that put us in a position of not knowing what it is we are being exposed to anymore. So thank you for your attention.
Cynthia, thank you so much for this fantastic talk. I really think you should go to schools and maybe you do and say voice that to everybody, because I think there is so much important information into what you just conveyed there. So we'll ask you a question in a little while in the Q&A session, but I just really want to mention, I suppose, that you really illustrate so well the concept of One Health. It's very powerful how decision that we make. And you articulated that really well. I think you mentioned how it was initially for the betterment of human life, right? We're trying to make things better, to make people have access to many things with wonderful properties, and then the damage that we make is so destroying. And the thing that is so powerful, I suppose, in your talk is that you show how we make a decision and then it impacts not only everybody everywhere on the planet, but also everything on the planet, going through soil, the air, and damaging every living cells there. So I think that was wonderful, yet a little depressing, but thank you so much for that. So we'll move on now to our next speaker, and we will definitely talk some more during the Q&A session. So now I am really happy to welcome John Mcginley. So John's background is in coordination and synthetic chemistry, and he has previously lectured in chemistry and in the Institute of Technology, Tala, and Maynooth University, both located in Ireland. So more recently, John has completed an MSc in Occupational and Environmental Health and Safety at NUI Galway, and is currently carrying out his PhD under the supervision of Dr. Alma Siggins and Professor Mark Healy at NUI Galway on the PestMan project, focusing on pesticide management for better water quality. So today, John is going to tell us about herbicides absorption by granulated activated carbon. Over to you, John. Thank you.
Okay. Hope everybody can see this. Thanks, Florence, for that introduction, and I'd like to thank you for this opportunity to talk today on the absorption of herbicides from aqueous solution by granulated activated carbon. So, as Florence said, this is part of the PestMan group, and PestMan stands for the pesticide management for better water quality. And so to begin with, I just want to give you a brief outline of what the presentation will be. There'll be some brief background, followed by experimental aims, methodology, results, and then some future work. So to begin with, what are pesticides? As you've already heard, pesticides are something that prevent, destroy, or control harmful organisms. or diseases, or they protect plants or plant products during production, storage, or transport. As the title of their talk here today is about herbicides, but the herbicides then they were developed to kill weeds. They're mainly used in farming, and they represent about 70% of all pesticides used in the United States, for example. So what were our experimental aims? Well, our experimental aims were, could we identify any media that would absorb pesticides? Could we then quantify their effectiveness? Could we identify catchment areas within Ireland that experience pesticide exceedances? And then place interventions in these catchments. So for example, here you can see this is a rather crude expression here, but this is the intervention that we've placed in the stream so that it will pick up any pesticides that are there. And then we would monitor their effectiveness. Another part of the PestMan project is to conduct the health risk analysis of the pesticide uptake by humans, but that's not going to be covered in today's presentation. So we started by looking at Department of Agriculture, Food and Marine reports on arable land and grassland and fodder crop production in Ireland. And these are the top 10 active ingredients or pesticides that are used for both of these based on kilograms. So when you can see here that MCPA has used 127,000 kilograms used of the active ingredient itself. A lot of these chemicals have, of course, been banned by the EU. So there's the ones with all the lines going through them. But at the same time, we then decided to go with the ones that I've highlighted in the boxes for use in our study. So we chose these 7 ones that you can see here on the right-hand side. Glyphosate occurs in both, as does fluoroxypride. Having chosen the 7, we ended up actually going with 6 because 2,4-DB has solubility issues, which we haven't realized at the time of our purchase. So what we did then was we looked for absorbent media. So the media was selected based on the criteria of low cost, bulk availability, and local sourcing in the Galway area. So we looked at a number of kind of industrial products, GSC, and then we also had 4 biochars that were available in the lab because of a previous project that were there. So we dried these overnight to 105 degrees, sieved them to get a size of about 1 to 2 millimeters, and then store them in airtight, sealable bags, sealable containers. But we just use little Ziploc bags, which is the handiest way that we discovered for this. In terms then of the herbicide concentration, we chose 100 parts per million, or that'll be 100 milligrams per liter, and made that up to using 0.01 molar calcium chloride solution. So what we did then was we took our media, took a media concentration of 5 grams per liter, took our solution of our herbicide and equilibrated for 72 hours at 10 degrees in amber glass bottles. And then we carried out our analysis. Now we chose to use a UV vis spectroscopy for the reason that it was really quick and readily available. The disadvantage, of course, is that you cannot get down to the nanogram per liter scale. But we felt that the speed of the reaction, or speed of being able to do the analysis, would have outweighed that. We also carried out at the time analysis using mass spectrometry, and the results that we got from the mass spectrometry mirrored what we were getting from UV vis spectroscopy, so we were quite happy with that. And these are the results that we see. So on the right-hand side of the screen here, you can see all the herbicides that we used, and then you can see the results as you can see. So you can clearly see that, granulated activated carbon, GSC, removed all the herbicides, bar glyphosate. But then this modified aluminium sludge that we had, that stepped into the breach and kind of removed all the glyphosate, or the majority of the glyphosate for us. But in that, and for the rest of this talk, we're just going to concentrate on what the GAC did for us. Oh, sorry. So these are the results of the kinetics that we carried out. Again, there were 100 mil or 100 ppm concentration and 5 grams per liter of the GAC, and we carried out the analysis from 0 to 24 hours. So as you can see, in the case of MCPA and Microprop P here at the top, most of the absorption was complete by 6 hours. Whereas in the case of 2,4-D, trichlopry and fluoroxypry, this was mainly completed after 18 hours. For the MCPA and the Microprop P, the kinetics of this turned out to be a pseudo second order, which was indicative of chemisorption being the rate limiting step. Whereas for the remaining three, 2,4-D, trichlopry and fluoroxypry, kinetics turned out to be pseudo first order, which again, it was indicative that physisorption was the rate limiting step in that particular case. We then carried out absorption isotherm experiments in these, again, across an absorbent range of 0.25 to 5 grams per liter for 75 or 72 hours, again, with a herbicide concentration of 100 parts per million. And I'm just going to focus now on the MCPA. So here you can see the linear and nonlinear various models that we chose. And in terms of the best R squared value or whatever that we chose to consider, Freundlich was always the best for all of the data that we chose. So our plan for the next coming months is that we've been currently looking at three catchment areas, three catchment here, Carduff, Dunleer, Craigduff, and a golf course in an urban area. And we've been monitoring the pesticide exceedances in these 4 different areas. So our plan is that we will put in our designed interventions in these catchments, and then we will monitor them to see the impact of those on the herbicide concentrations over that year. With the website and Twitter account are shown here for Pestman, the Pestman project. So it's pestman.ie, or the Twitter handle is here if you're interested in following to see how we get on. And finally, I'd like to thank Professor Mcginley and Dr. Alma Higgins for their support and encouragement over the last number of months. Liam Morrison and Porrick Ryan and Perrick Malander for their advice and encouragement. They're also part of the Pestman team. I'd like to thank, wouldn't like to thank, but Joni Mitchell has been an inspiration. I'd like to thank also the EPA and the Department of Agriculture, Food and Reed for sponsoring this. And finally, I'd like to thank you all for listening. Thank you.
Thank you very much, John. That was a really an enjoyable talk. So please do put your question in the Q&A box, and then John, we will come back to you in a little while when we do our Q&A session. So thank you very much for that. John, so now I would like to welcome Gaurav Chu, who actually is from my lab, where he's carrying out his PhD under my supervision. And Gaurav is also co-supervised by Professor Conor O'Byrne from microbiology here at NY Galway. So Gaurav's research focuses on Pfas. So Cynthia mentioned them. So they're, I suppose, pollutants, organic contaminants. And what Gaurav is looking at is the effect of Pfas on microbial cells. So Gaurav obtained his BSc and his MSc in botany from the University of Delhi in India. And after that, he worked as a project associate at the Terry Dinkin Nanobiotechnology Center. And there he investigated protein-mediated biosynthesis of zinc and iron nanoparticles from mining wastes. So today, Gaurav is going to talk about microbial responses to organic pollutants. So over to you, Gaurav.
Thanks, Florence, for the introduction. So hello, everyone. Hope you can see my slides here.
Yes, we can.
So Perfect. So I'll be talking about my research on microbial responses to organic pollutants, particularly PFOA and octanoic acid. So as it has been already introduced by Cynthia, but I will still take you through and let's briefly talk about Pfas or Pearl and polyfluoroalkyl substances, or as they are infamously known as forever chemicals, because they have the tendency to persist and bioaccumulate. Pfas are man-made chemicals that repel oil and water, and there are around 3,000 plus Pfas compounds known to date, among which PFOA is one of the most widely studied forms of Pfas. These compounds are used in a variety of consumer products that we use on a daily basis, such as non-stick cookware, water-resistant boots, aqueous film-forming foams, waterproof clothing, and our pizza boxes, and also our microwavable popcorn bags. So being present in all these products, we can assume that these PFAS compounds get into the environment and pollute water, soil, and air, and it ultimately affects us as well. And also various health effects have been shown to be associated with the exposure of PFAS, such as disrupt thyroid function, reduced response to vaccines, osteoarthritis, immune response suppression. But most alarmingly, pregnant women, unborn fetuses, infants are most susceptible to the adverse effects of these PFAS, causing low birth weight, difficulty in becoming pregnant, preterm birth, and also attention deficient or hyperactivity disorder. Although various health effects have been associated with PFAS exposure, but the mechanistic basis is still unknown. And here we hope to get a handle on the mechanistic aspect of these compounds on living cells using microorganisms as model. So let's look at how PFAS impacts microorganisms. These toxic organic hydrophobic chemicals target cytoplasmic membrane, causing membrane disruption and ultimately causing cell inactivation and death. PFAS, particularly P4, acts as a weak acid and exists in a pH-dependent equilibrium between dissociated and undissociated forms. So here, particularly the undissociated form enters the cell, disrupting the homeostasis of iron and solutes, reducing the intracellular pH, which ultimately impacts the ability to tolerate stress, which could be osmotic or acid stress. So the overarching goal is to understand mechanistically how P4 affect microbial cells, and we are taking E. coli as a model organism. We are studying the evolution of E. coli in presence of P4, for which we follow the in vitro evolution of the strain in presence of this compound. And also in parallel to PFOA, we are using octanoic acid as a control, as it is also a weak acid. It has the same C8 structure, but without fluorine. So ultimately, we are going to look at the genetic aspect of how E. coli is impacted in presence of these compounds. Additionally, we are also going to look at the transcriptional response of E. coli to these compounds to look how the organism is responding. And we believe that combining those two sets of data, we hope that we'll be able to put together a mode of action of how this compound is affecting. But for today's talk, I will only be focusing on the in vitro evolution experiment here. So in our experiments, we have seen that PFOA at 500 mg per liter concentration does not affect the growth of E. coli at normal physiological conditions at pH 7 in M9 minimal media when supplied as a stressor with glucose as a carbon source. But PFOA and octanoic acid both at 500 mg per liter concentration appears to impact the survival of E. coli at pH 3 after 5-minute time point. And this phenotype provides us the opportunity to study the effect of these compounds that have on E. coli at low pH. So further, we found that at pH 5.2, PFOA and octanoic acid impact the growth of E. coli. And here we did a broad range finding to find a suitable concentration for PFOA and octanoic acid. And we found that at PFOA concentration of 1.4 millimolar and octanoic concentration of 0.2 millimolar, it gives us the same growth rates for both, and this is good enough for us to follow through the in vitro evolution experiment. And also the ratios of the acid and the conjugate base for these two weak acids justifies the same response in growth rates to the difference in the concentration used. So further on, I will take you through the setup for the in vitro evolution experiment. E. coli was used as a model and grown in M9 minimal media at pH 7 and was further inoculated in 4 conditions with 3 tubes each. 4 conditions being M9 at pH 7, M9 at pH 5.2, M9 at pH 5.2 with 1.4 millimolar PFOA, and M9 at pH 5.2 with 0.2 millimolar of octanoic acid. And 1:100 passage was done every day for 37 days with the final OD measured, and the stocks were made with 7% DMSO, which is a cryoprotectant. The stocks were made maintaining the same pressure as they were in the in vitro evolution experiment, and they were stored at -80. Also, the growth curve was performed every 6th day and the growth rates determined. Here I show you the overall summary of the results for our in vitro evolution experiment with the final ODs plotted for all the conditions every day. Here for the octanoic acid samples, I would like to draw your attention that we see that after 12 days, there is an increase in the final OD observed. And also at day 17, we see the highest final OD. And if we move to the pH 7 samples, we do see an increase in the growth in the final OD after day 6. But in our growth rates, we could not see any change, and the growth rates remained the same throughout. Similarly, for pH 5.2 samples here, no change in growth rate was observed throughout the experiment. And for PFOA samples, they did start together with the octanoic acid, but then they diverged. And after that, we see there is no change in the final OD or growth rates, and it remains unchanged. So now I'll be focusing on the octanoic acid passages. So here, now focusing on octanoic acid passages from day 9, the growth advantage from day 9 is seen as compared to the parent in all the 3 tubes. And I would like to tell you that we chose to focus on day 9 instead of 12 was to be able to catch the change as early as possible in the passage to avoid any noise in the mutation we may see. And as previously, we have seen that we observed the increase in the growth rate that happened between day 6 and 12 from the growth curves that we did every 6th day. And hence we decided to look at a day that was in between so as to catch the mutation as early as possible. So day 9 gave us the same phenotype, and we chose to follow day 9 further on. So similarly, increased survival is also seen for day 9 octanoic acid passages at pH 3 when compared to parent. Here we can see the increased survival even after 10-minute time point for 2 tubes out of 3 tubes. And also at 10-minute time point, we can see there is an increased survival when compared to the parent. So, which clearly shows that there's an advantage to these passages than the parent. Hence, the next step was to isolate the colonies because we cannot assume to have clonal population in every tube. Hence, we isolated the single colonies, and these were screened for the evolved phenotype, and all the colonies demonstrated the same evolved phenotype. Further, 3 colonies from each passage was isolated, and then those were to be sent for the sequencing. And to tell you about the work apart from this, which I have shown, and as I have previously shown you, that we observed that the highest OD for the octanoic acid passages was seen at day 17. So we chose to look at that as well as at day 37 passages, so as to see if they still remain, to see if they still retain the phenotype towards the very end of the experiment. And thankfully, we found that for passages 17 and 37, they display the same phenotype as day 9, the evolved phenotype. So further on, we plan to perform the same studies for passages from pH 7, pH 5.2, and pH 5.2 with PFOA. But the problem we are faced with is we are able to revive the stocks that we made for pH 7 and pH 5.2. But for pH 5.2 with PFOA, we are not being able to revive the samples, the stocks, when we maintain the same pressure, that is pH 5.2 with PFOA. And as I previously told you, that all these stocks were made maintaining the pressure that they were undergoing in the in vitro evolution experiment. And we revived them with the corresponding pressure. So here, for example, for the PFOA samples, they were everyday passaged in the M9 minimal media with pH 5.2 with PFOA, and then they were stored with the same pressure. And then when we are reviving them, we revived them in the same M9 minimal media with the same PFOA pressure, but they are not behaving well there. But we are able to revive them at pH 7. So there must be something happening there. And this happens upon freezing. So believe that they cannot tolerate the low pH or PFOA stress upon freezing. And further on, so the next step immediate is to select the colonies for the other 3 conditions, as I told you, and send them for sequencing. And we plan to do comparative genomics. And as I mentioned previously, we plan to do comparative transcriptomics on the cells prior to in vitro evolution. And we are hoping that we might see an overlap in the gene that have mutated with the gene that were differently expressed before. So to conclude, we have seen that PFOA and octanoic acid impact the growth of E. coli at pH 5.2. PFOA and octanoic acid impacts the ability of the strain to survive the stress at pH 3. And we have seen E. coli has shown to evolve and show increased growth and survival for phenotypes for octanoic acid. And also the isolated colonies from those passages show the same evolved phenotype. And we are facing problems with reviving the PFOA stocks, which clearly evidence that fluorinated compound is affecting E. coli differently from the non-fluorinated forms, because we are able to revive the samples for the octanoic acid, but for the fluorinated counterpart, PFOA, they are proving to be difficult. So this needs further investigation, and I would like to thank Florence Connor for their supervision and support, and also for the encouragement for my lab members and my GRC panel, and funding from Hardiman and Thomas Crawford Hayes Research Awards. Thank you very much. Thank you for listening.
Thank you very much, Gaurav, for your presentation. So again, please don't hesitate to put your question in the Q&A box, and then we will have a conversation then all together with all of our speakers after our final talk. So I am absolutely delighted to introduce Dr. Allison Connolly. So she's currently an Irish Research Council Marie Curie Action Postdoctoral Research Fellow, and she's conducting the IMAGE project, focusing on Ireland's biomonitoring assessment of glyphosate exposures. So Allison's research of interest is on exposure assessment studies, and particularly looking at human exposures to pollutants using biomonitoring sampling strategy. So she's also interested in both environmental and occupational exposures. So Allison is currently on the board of the European chapter of the International Society of Exposure Science as a newly elected counselor of exposure science capacity building, training and education. So today Allison is going to talk about the IMAGE project regarding glyphosate exposure. So it's a pre-recorded talk that Allison recorded for us, but she's here with us, so she will be able to answer all of your questions at the Q&A session. Okay. So just bear with me 1 sec, so I just get her video started, and here we go.
My name's Allison Connolly, and I'm here today to talk to you about my research project, IMAGE, Ireland's biomonitoring assessment of glyphosate exposures. And if you're on Twitter, my handle is @AllieConnolly18. So just to give you an overview of our study, the aim of our study was to evaluate glyphosate exposures among farm and non-farm families using a human biomonitoring approach. Glyphosate is a herbicidal product that's used in gardens and in agricultural settings, and is most well known as the Roundup products. So why do we evaluate glyphosate? Well, there has been health risk assessments of glyphosate. So the International Agency for Research on Cancer classed as a 2A, probably carcinogenic to humans. However, other agencies have had differing classifications. So the European Chemical Agency, the European Food Safety Authority, and the US EPA have differed in their classification. And currently it is under review in Europe by ECHA and EFSA. But the important thing with this chemical is that it is the highest volume used herbicide in the world, in over 750 products, and we do need more exposure data on this chemical. So how do we evaluate pesticides using a human biomonitoring approach? Well, the first thing we consider is the exposure source. So in this case, it could be from agricultural settings, from farmers spraying crops, or else it could be from a secondary exposure, like if you live near a farm setting. Other sources are if you spray it yourself, so you might use it in your own garden, or if you ingest it. So this could be from contaminated water or from food sources. So crops, wheat, potatoes can be sprayed with pesticides. And then dust. So if you live near a place where it's sprayed with pesticides, there's potential to be in dust within the home. So then we have to consider the exposure route. How do people actually absorb it into their body? And it can be inhaled. So if you're spraying it, you could inhale it. It can be ingested. This would usually be dietary, ingesting water or food samples that are contaminated with pesticides, or from skin uptake. So the dominant route of exposure for spraying is actually absorbing it through your skin. Once it's in the body, we have to consider the toxikinetics. How is it absorbed? How is it distributed throughout the body? How is it metabolized? So in this case, glyphosate is mostly metabolized into, and only a small fraction of it is metabolized into AMPA, amino methylphosphonic acid, and then it's excreted. And this is where human biomonitoring comes into play, because we can collect these urine samples and then detect levels of the pesticide compound, so glyphosate in this case, or its metabolite, so AMPA. And with this information, we can use it to evaluate the exposure and risk, which then feeds into our exposure management plan, considering the routes and sources of exposure and trying to control it. So what have they found on glyphosate so far? So a recent review study that we conducted, we found that there's been studies done all over the world, and what they do find is that it does seem to be ubiquitous in the environment. However, there's very little data on families' exposures. So this is something we do need to investigate further. And there's a link here for anyone that's interested in this article. So IMAGE is looking at families' exposures in Ireland. We had 14 farm families and 53 non-farm families participate in the study. We asked that both parents and at least one child participate in the study. If people had more children, we would collect the samples as well, but that was up to the parents. But we needed a minimum of one child. And we asked that at least one child be between the ages of 6 and 17 years old. What kind of samples did we collect? So we collected the one full void urine sample from each participant. So this means that you collect the whole sample, and we collect it first thing in the morning. We also collected additional samples. So we collected water samples from houses throughout Ireland, and we had a smaller study looking at dust samples. So this was with farm and non-farm families. And from all the families, we collected contextual information. So this was questionnaire data that was designed by the Human Biomonitoring for Europe initiative. So what we found. So just to give you an overall of the results, this is both farm and non-farm family. We found that the frequency of glyphosate was 26% of the samples had detectable glyphosate levels, and 59% had detectable AMPA levels. So the breakdown products seemed to be more detected than the parent compound in this case. The levels were at max level of 3.21. So to put this into context, when we did an occupational study in Ireland previously, our max level was 10.66. And when there was a farm and non-farm family study done in America, their max level was 9.4 micrograms per litre. So this is similar to what you'd expect. And the medium levels were below the limit of quantification or near the limit of quantification. In this case, for our study, it was 0.05. And they were quite low concentration values we found. So when we looked at these urinary results, when we took farm and non-farm families, you can see here in the non-farm families that if you look at glyphosate in both, it had a similar frequency of detection. The max value was similar. The only thing that was slightly different was the 95th percentile. And then again with AMPA, the frequency were nearly the same, as were the max values and medium levels. And there was a slight elevation actually in the non-farm family for AMPA. But again, these were all quite comparable results. However, when we looked at the frequency of detection, when we split it from mothers, fathers, and children, we actually found that the highest frequency was in the children, which is in the blue bars, followed by the fathers, and then the mothers. Now, other pesticide studies, including glyphosate, have found higher glyphosate levels in biofluids in children than in their adult counterparts. So this is not unusual. And the concentrations were low across all the participants. So what does this data tell us? Well, overall, if we look at these concentrations using the European Food Safety Authority's acceptable daily intake allowance, so you can see the equation here, the percentage of an ADI is taking your glyphosate concentration in urine times by the volume of excretion of urine, and you divide that by your body weight, this urinary excretion fraction. So what goes into the body and what gets excreted out of the body is not always excreted out of urine. So we usually have a correction factor for this, and EFSA have put this at 20% that is excreted via urine. And then you times that by the ADI of the chemical you're looking at. And what we found here was our max values was only 2% of the ADI for glyphosate and only 5% of the ADI for AMPA. So you can see that they are low in comparison to our guidance values. And we did find a higher frequency of AMPA versus glyphosate. So glyphosate, only a small fraction of glyphosate breaks down into AMPA in the body. However, it is an environmental breakdown product. So if you spray it on crops, for example, it can break down in the soil and leach into water courses. So this potentially could be exposure from environmental factors like ingestion of water or intake of water. So what are we going to do next? Well, the next steps of the study is we want to investigate this urinary data further. Unfortunately, we've only had a preliminary look at the data, so we haven't been able to investigate it in depth. We also want to look at the water and dust data. So we're still waiting on the analysis of this data. So that will be interesting to identify the sources of exposure and to look at that alongside our human biomonitoring data. And then to look at our food diary and contextual information. So again, we collected a food diary of what they ate 48 hours before they gave a sample. And we also collected contextual information like questionnaire information. And that we can use then to identify potential sources and routes of exposure. I would just like to take a moment to thank my research team, who have been amazing, and for the funders, the Irish Research Council, Marie Curie Co-Fund. It's been a phenomenal experience. If anyone has any questions, please ask now, or else my email address is up here, allison.connolly@nuigalway.ie. Thank you.
So thank you very much, Alison, for this wonderful and very clear talk. So I think, and thank you to all of our speakers right before we start the Q&A session. So please, if you have any questions, make sure to put them in the Q&A box. So we have a couple, but I think I might just start, if that's okay, by making, I suppose, maybe a comment on Cynthia's talk. So one of the things that strikes me, with all of the data that you presented to us is a lot of people think they're very healthy by, let's say, eating lots of fish. You see that a little bit. And one of the things that worries me a little bit, so I've noticed that in some of the questionnaires that you might have, when you do, when you look at human characteristics, you might have a threshold where if people eat more than twice a week fish, they're going to be put into another category because we know they might have more bio burden, I suppose. So I was just wondering, how do you think it's adequately, this message is adequately disseminated in a way? Like, are people aware that by trying to be healthy, let's say by eating fish, they might actually be more exposed, it might be detrimental to their health? Do you have any comments on that?
I don't know. It depends. I mean, there are different fish species, and this is something we found in Sweden, is that when they try to give dietary advice to people, that people don't know the difference between salmonid fish, which are the fatty fish like salmon and trout and herring, and the very lean fish, which are white fish, for instance, cod and plaice and the flatfishes and so on. And because the flatfish and the whitefish are very lean, they have very little fat. So actually they're good for you. You should eat a lot of fish in that respect, because they have since they have very low fat contents, they have very little of these contaminants in them. And it's mostly these fatty fish. And I mean, the herring in the Baltic Sea are still not allowed to be sold within the European Union because the levels of dioxins are still quite high. They're going down, but they're still high enough that the EU does not want them used as food for people. So that is one way you protect people, is you just say you can't sell this on the market. Then people don't have to know. But when it comes to helping people choose, it's much more difficult. I think the food authorities all find this a very difficult task, to let people know what they should and shouldn't be eating. And it's not just pops, it's heavy metals. Mercury is a typical, it used to be you were not supposed to eat tuna fish, for instance. And I still think tuna fish still has high mercury levels. So, it's the same thing. It's hard to reach people and help them choose properly.
Yeah, I think it's a very complex question for sure. I think, and also, you mentioned people might not have to know. You always want to make sure you don't make people too concerned, and at the same time, you should really disseminate that knowledge for people to try to make informed choices, and also, maybe try to change their patterns, because obviously that we have something has to be done. So I have plenty of other questions, but there is questions from people, so I have to to also honor their questions. So I have a question here from Alma Siggins. So considering chemical compounds are being introduced to the market at a higher rate than ever before, is there a case to be made for industry to play a part in regulation, or better still, remediation process?
Well, when it comes to playing a part in regulation, to some extent, this is already happening in REACH, at least in the sense that when a new chemical is to be put on the market, the industry has to actually test it and provide the test results to ECHA, who can then try to do a risk assessment and decide whether or not this chemical is toxic or a potential environmental contaminant. Unfortunately, the chemicals are being produced faster than ECHA has a chance to evaluate. So I think sometimes they have let things on the market and hope that the results that they companies are giving to them are correct. So yes, there is some part to play in regulation. When it comes to remediation, this is a bit trickier because here you have the problem with whose fault is it? And we see this now with the perfluorinated compounds, Pfas, that the companies that have produced these chemicals and released them to the environment, and in many cases legally dumped them, are doing everything they can to fight any kind of legal actions that would force them to stop producing or to clean up these sites. But I think many of them do take some responsibility and have been trying to clean up. But you have old sites as well that there's no one left to take care of them anymore. And then it falls to governments to do this. So I think, yes, I think it would be very good if industry would take more responsibility for what they're actually producing. And actually, I think it would be better even to go the other way and go over to what is sometimes called sustainable chemistry or green chemistry, which is producing chemicals that break down in the environment to harmless compounds. They call it design, but benign by design. So creating chemicals that actually don't cause problems in the future.
Yeah, that seems definitely like that's what we need to enforce, because one of the things you mentioned as well is how every chemical seems to be banned and then replaced with something almost worse than what we had before. So we are making the problem, even bigger in a way, because now we have to tackle many different targets, I suppose, as opposed to have, maybe fewer. So one final question for you, because I'm aware that we have also other questions for the other, and we might come back to you if time's allow after. But Gavila is here, has asked us so what is the typical size of the particles that can transmit POPs in air, such as those detected in the high Arctic? And she would like to know if there are any reports of pharmaceuticals being transmitted in a similar way.
Okay, I'm not sure exactly of the size of the particles. The atmospheric science people would know this, but they're the size particles that basically are small enough to be in the air for a long time, so they don't fall out of the atmosphere as easily, because big particles will fall, but small particles will go up. But then they are also big enough that eventually they do slowly possibly drop out. But you also have these condensation, that they will condense onto water droplets, and typically the rain or in snow. So as soon as it snows, these can be condensed out, and then they snow out onto the ground. The pharmaceuticals are an interesting question. I have not heard of anyone finding pharmaceuticals in air samples. However, they are found in water samples. And in the Arctic, they have mostly been found near settlements, and they think they're coming from the sewage outfalls. Around small towns or villages, because in a lot of places in the Arctic, you do not have sewage treatment plants. So sewage goes straight out. And where you do have sewage treatment, it's very often very simple sewage treatment. And pharmaceuticals are water soluble, so they often will go right through a sewage treatment plant anyway. So they are actually reaching the environment probably from local communities.
Yeah, thank you so much, Cynthia, and yeah, thank you for those questions as well. So I might ask a question to John. John, so you mentioned you're here, John, are you? I can't really see you. So you yeah, great. So John, you mentioned, and I think it's a great idea, and I totally see the rationale for it. But you mentioned that you're looking at waste material, right? And then you're trying to see if they can absorb so pesticides, and you source them locally. And I totally see the reason. I think that's very important to do that. But does this mean that you have to deploy the same experimental strategy in different geographical locations due to the fact that you want your waste material to come from where you're carrying the study?
No, our idea was that we would take some materials and see which of them would absorb the herbicides best or the pesticides best. And from the results of our experiments, like GAC it was miles ahead of anything else. So granted, or like if there was some other material that we hadn't tried, we would try that. But at the moment, we're just happy to go with the GAC. Like it's one of the issues with GAC is that it's not low cost. So that's a different thing. So, but at the moment it's GAC or in the case of glyphosate, it's the modified wastewater sludge. So yeah.
Great. So I suppose one small follow-up question, if that's okay. So I was wondering if there is any way to predict the absorption you might expect. So you show us you have different kinetics, and they must be dependent on both the characteristics, physical, chemical characteristics of your your media and then your compound as well. And I was wondering if there is any way to become predictive or to try to use all of the data you can find and try to develop models.
We were one of the things we were surprised at was that the biochar actually didn't absorb any of the pesticides that we were looking at, or the herbicides, because we would, based on some previous work by Alma and I think yourself, that it showed that TCE, for example, was absorbed pretty readily. So we thought that biochar would be good. So from that point of view, then you can't really predict. It presumes that the pore volume and the pore size would be somewhat similar between GAC and biochar. So we thought, okay, we're with a good shout here, but as it turned out, biochar really didn't do a whole lot. So yeah. Yes, I hope that answers your question.
It does, yeah. So there is a question from Allison, who just gave her talks there. She wants to know if you measure, because of course she's working on glyphosate as well, right?
Yeah.
So she would like to know, and I feel very strange to ask your question because you're right here, Allison, but did you look at AMPA, the degradation? No, you just look at that.
No, we're just, at the moment, we're just concentrating on herbicides at the moment. We're not looking at the metabolites at all. No.
Okay, great. And we know from Cynthia's talks that the metabolites can go.
Sure, yeah.
That as well.
But at the moment, we're just concentrating on that, and I presume it might be a follow-up project or something like that. So,
yeah, sure. Thank you very much. I might just move on to Allison then, and we'll have, of course, I won't forget you. But Allison, so one of the things I was, I felt very sad about, again, I suppose, is to see that the poor children are the one with the worst bioburden. And you said that that's actually common, and I was wondering why. Do we know why that is the case?
So it depends sometimes. It could be biological, the way they break it down, or it could be from sources. So especially in farm families, a lot of kids like to be out playing in the fields and stuff like that. So it's very preliminary what we've done so far. We only got the results a few weeks ago, but we would be interested to know, because we did see that there was a lot of people living near farm areas. So it could be a potential of secondary sources from being outside playing in the areas. But without investigating further, we don't know whether it's biological or is it from additional sources. But it could be secondary from being near farmland.
Yeah, that kind of makes sense, I suppose. So I also wanted to ask you, so how does glyphosate get to AMPA? Does it involve, let's say, obviously I'm a microbiologist, does it involve any microbial enzyme? Can a microorganism do that? Or how, how is it mediated, I suppose, that transformation?
So it's actually a really good question, because in humans, it actually only a fraction of it breaks down into AMPA. So when we're measuring for, let's say, intake, we might measure AMPA and glyphosate, but very little is transformed from glyphosate to AMPA in the human body. But when it's sprayed, let's say in the fields, it actually breaks down in soil and then can leach into water and that. So that's actually how it breaks down. So really, if you have a high AMPA level, we assume it's from secondary sources. So drinking water would be one of our
like main assumptions because of it is detected in water or a breakdown on food stuff. So it is it's an interesting question. It does kind of highlight for us in our study that we had higher AMPA levels that is potentially not from intake of glyphosate so much as intake of AMPA from different sources.
Thank you very much, Alison. I'm so sorry, we're kind of running out of time.
Okay.
So I have just one question for you, Gaurav. What do you think is going on with your PFOA cells? Why won't they revive, do you think? Why do they refuse to take any? They're alive, you know, they're alive after freezing, but they just won't grow for you if there is any PFOA or if there is any acids. Do you have any, very briefly, do you have any insight into what could be the case? What could be going on?
So like I would say, like as we have seen that previously, like PFOA appears to have a different effect on microbial cells, like especially after freezing. So so I assume that we are using PFOA at a very high concentration, and PFOA specifically is known to disrupt the membrane function. And it also affects the lipid or fatty acid beta oxidation. So I assume that there's some role that's playing that it is getting too much into the microbial cells through the membrane because the membrane function is disrupted. So at this particular high concentration, it's causing much more lethality than the octanoic acid, although we see the same effect in the growth rates, like the decrease in the growth rate, but in terms of the internalization of the PFOA into the cells, I assume that's hampering the growth of that's like hampering the survival of those cells, like, you know, especially after freezing, because that's an additional stress to them. So I think that's something to do with the mode of action of PFOA, that's the fluorinated compound and the non-fluorinated. So it would give us a great insights, as I mentioned, to see the difference that they create the fluorinated and the non-fluorinated compound. So like once we have like the sequencing, we would have further insights into the differential effect. So let's hope for the best.
Great, thank you so much, and I'll be well aware of what's going on, hopefully. So I want to thank absolutely everybody again for the wonderful talk. I really enjoyed the session, and I hope you have too. Thank you everybody for joining us. And so there is one final session then coming up, which will focus on people, nature, and the environment, and will be chaired by Dr. Maria Tui. So please join us at 12:30. So in about 15 minutes for the final session. Thank you everybody so much, and particularly to our speakers. Thank you. Bye-bye.